Good morning, good afternoon, who can join. I'm happy to be here at the Symposium of the Spinal Cord Injury. My name is Dr. Lior Shaltiel. I am the CEO and Director of NurExone Biologic, and I'm pleasure to be here and present some of our work. We will talk also on the clinical development of the company and how we're transforming the regenerative medicine space using exosome loaded with siRNA. We are a publicly traded company, and this is the reason that we have a disclaimer right in the front as a second slide, but we can move on to the agenda where when I will present the overview of the company, we will talk about exosomes and how they are the ultimate drug delivery system. We will speak about our product, XOP10, our lead indications for acute spinal cord injury. I will show you some preclinical results and our path to a clinical stage company for a phase 1/2A with spinal cord injury. Our mission is very simple. We want to develop a regenerative medicine product to target nerves and recover the injury after spinal cord injury or different injury in the CNS, central nervous system. And we are talking about three elements: the regeneration, rewiring, and recovery, functional recovery of the patients. Our lead product is XOP10 and is target neuron relative specifically, showing motor, sensoric, and functional recovery in our animal model. We founded in 2020 after several years of joint research between two universities, the Technion and Tel Aviv University. Professor Shulamit Levenberg joined forces with Dr. Danny Offen from Tel Aviv University, working on spinal cord injury with different approach. The best approach shows that exosome loaded with a P10 siRNA showed the promising results in an intranasal administration. Well, this is the reason that the company established. We also successfully made an RTO 3 years ago to Canada. So we are a publicly traded company under the symbol NRX. We are traded also in Germany, both in Germany, and also in the US in the OTCQB under the symbol NRXBF. Our main assets are strong patents, 5 families of patents. 2 of them are a worldwide exclusive license from the Technion and Tel Aviv University. We have orphan drug designation in Europe and United States, allow us to accelerate the regulatory approval and getting the significant assistance for the agencies. We have secured the master cell bank to allow us to produce the exosomes from from the best raw material that we can get. This is a full GMP characterized, FDA approved bone marrow mesenchymal stem cells. And so far, we raised $17 million in the last 4.5 years, when the founders are still keeping 25% of holding in the company. The structure is the company after the RTO is the mother company in Canada, holding 2 subsidiaries, one in Israel, the R&D to produce the XOP10 and other pharmaceutical molecules, going all the way with the FDA and EMA approach. And the second one is a US subsidiary established this year called Exotop, where we were gonna produce from our master cell bank the naive exosomes, our DS drug substance for our product, and also have the ability to supply the intensive demands for naive exosomes for different markets such as aesthetic, cosmetics, and other private clinics in the field. Those are our patents. We have 5 families of patents. The first one is the drug administration, talking about XOP10 in intranasal, and also different other administration routes dealing with specific neuron damage. So it's not only spinal cord injury. It's granted in the US, Russia, Japan, and Israel, and we are pending for other territories. The second one contained 2 PCT patents regarding the production and the composition of matter of exosomes. We are talking about moving from 2D to 3D productions with higher yield and increased potential efficacy of the exosomes. We are talking about also what we load inside. So we also patent the siRNA sequence and modification of the sequence, not only for exoP10, also for other 2 proteins we target in other products. And the last one, and I think the most important one, is our exosome loading technology. We are talking about loading post-production of the exosomes with our siRNA. There is no gene modification of the cell source. It's not toxic, and we even can tune the efficacy of the loading by adding non-toxic third molecule in order to push the loading to a higher grade. Now we talk about what we address. The first one was acute spinal cord injury. And the second one is glaucoma or acute glaucoma. Both are the same mechanism of action. We are talking about acute phase of injury of the CNS, central nerve systems. Both markets are relatively large, and that we are targeting $1 billion markets. Today, we will focus solely on the spinal cord injury, and I will show you results on that. Exosome, why we use exosome? What are exosomes? Exosomes are tiny, nanoscale, bilayer vesicles secreted from cells, in our case, bone marrow mesenchymal stem cells, and they have amazing nature. They are homing from a healthy tissue to a damaged tissue in order to communicate cell to cells. We're using it for our advantage, and we getting the exosome accumulate in the injury by the fact that the exosomes themselves go into inflammation and accumulate there. And they are not just a drug delivery system, they are drug delivery system with a therapeutic cargo, okay? They have their own therapeutic effect, and what we see is a synergistic effect between their therapeutic effect and the siRNA. So we push the system towards regeneration. exoP10 is a loaded exosomes. So we have the exosomes and we have the siRNA P10. The idea is to deliver it in intranasal or intrathecal administration, homing to inflammation, and we can see they are targeting and engulfed by nerves, okay? And the P10 is in the mTOR single transaction, and we actually inhibit the inhibitor. When we inhibit the inhibitor, we push the system towards regeneration, and we can see exon growth. This is our scheme of models. We use, as you can see down, 2 different models: full transection, okay, taking the spinal cord and cut it completely, or compression. In both cases, the structure of the preclinical test is relatively the same. We have the injury, we have the treatment, can be during the first or the second week. Okay, we are training them with treadmill daily. We're taking BBB. The BBB is the scale for motor function. We also Wonfray for sensoric. We weighting them on a weekly basis. And in the end, we have MRI scanning and histology. And some of the results I will show you today. But let's start from the video. You can see on the right, this is a rat after full transection. Got intranasal administration. And we can see even after 2 weeks to 1 month that the rat on the right could walk. Walk awkwardly, but he walks and he put the legs and all the weight on the back legs. On the left side, those are the control rats. Here we are talking about full transection using a PBS, and you can see that he is actually carrying his leg, completely paralyzed, and this is very stable. There is no functional recovery, spontaneous self-recovery after GHG, which we usually see in other models. So we took this transection model, and we see here on the top left the BBB. Of course, the BBB in this case is lower than what we see in the compression. In the compression, we can push it even higher than 4. And in the last results, we even can push it to more than 10. Okay, and we can see that it's a synergistic effect and significant results when we use 4-time exopitan. We also add the bone fray for sensoric, and we can see that 75% of the rats would feel again the legs. And this is also reflected in the quality of life. Rats after treatment, in our case, are not prone to eat their legs due to the fact that they probably feel. In cases of control, we see self-mutilation of the legs due to pain, phantom pain, in this population. So this is also a very positive remarks to say. We also measure the gray matter, and here is also significant results showing increasing in the gray matter, meaning that we can rescue more tissue in the spinal cord injury after the injury. And we take these rats after the 2 months of study, and we take the MRI DTI in order to see the tissue itself and the direction of the water in the injury. And you can see on the top left, sorry, the injury, and this is a full transection. You see the degeneration of the nerves after the injury, while in the treated one, we can see recovery of the tissue, reconnectivity, and even to get it to a normal value like we saw before the injury. So the width of the spinal cord injury is not damaged, and this is also one of the reasons that we can see regeneration and functional recovery. Moreover, we research also what's going on with the blood flow, blood vessels, and we can see very significant results showing that in the area of the injury after treating with exopitan, we can see increase in the blood vessels area, and although the numbers are staying the same, the numbers, the blood vessels area in the area of an injury are higher in exopitan, meaning that we have more blood flow into the region, more probably demand of blood. That's meaning that we have more cells in the damaged tissue, injury site. This is a comparisons between different competitors. We have different approach how to treat spinal cord injury. And you can see here that we tick all the boxes. First of all, one of the only one, I think, that we show full transection model, okay? And this is the most severe model. It's a 0-1 model. If it works, perfect. If it doesn't work, that means that there is no regeneration. The technique is non-immunogenic, so there is no toxicity to this. It's very natural. We can use it off the shelf. So one batch for 7 patients is totally allogenic approach, and we use it in a minimally invasive. And I think this is the key point. We are not talking about invasive approach. We don't need another operation in the spinal cord injury. This product can be delivered intranasally or intrathecally into the site. So first in human, how it will look like. The primary objective is, first of all, to deal with patients with acute spinal cord injury. We are going to evaluate also the neurological function following the administration of exopitan, and we are targeting acute spinal cord injury 3 to 7 days after the injury. So in an acute phase, we are aiming between C6 and T10, and we want to push patients with Asia A towards Asia B. That's practically what we will do. Due to the fact that I don't have a lot of slides, so I will move on very quickly. This is the part 1. It will be phase 1, 1 arm, open label. We will have up to 18 patients. We are doing dose escalation with that, and we will monitor the population for safety. The second part, it will be randomized, double-blinded, another 10 to 15 patients, okay? And we want to show dose, we want to define the dose that is relevant and the most effective results for the XOP10. And that's the summary. I can show you the platform technology, our XOP10. We're talking about two different indications, spinal cord injury and acute glaucoma. We have a hybrid model of combining pharmaceutical company that develop XOP10 for clinical study and Exotope that also producing a naive exosome for also have a cash revenue producing during the process. And we are currently seeking for a strategic partner to taking us to a full IPO in the US major markets, like NASDAQ and New York Exchange. And with that, I'm going to finish, and I would like to say thank you for your attention, and I'm looking forward to stay in touch with you. Thank you.